Hello everyone. In this module, we shall study the development of the female reproductive system in plants. The floral meristem terminates by forming the carpels. A carpel encloses the ovules in angiosperms. In gymnosperms, the ovules occur as naked structures, often borne in the axils of leaf-like organs. The carpel confers a number of advantages on the flowering plants. It protects the ovules within them. It enables pollen capture and guides the pollen tube to the ovule. After fertilization, the carpel tissues undergo developmental changes to form fruits which protect the developing seeds and contribute to their dispersal. The learning objectives of this module are to study how the carpel develops and forms the syncarpous pistil in Arabidopsis, which bears the ovules. Then, to see how the female gametophyte and the egg cell is formed within the ovule. And finally, we will try to understand how these developmental processes are regulated at the molecular level. As in the earlier modules, we shall restrict our studies on carpel development to Arabidopsis. In Arabidopsis, two carpels are joined along the apical basal axis to form a pistil, which is attached to the base of the flower by a short internode called the gynophore. The individual carpels and the syncarpous pistil are divided from the apex to the base into tissues which perform distinct roles in reproduction. The stigma at the apex is specialized for the capture and germination of pollen grains. The style conducts pollen tubes to the ovary within which lie the ovules. Each carpel is thought to have evolved from a leaf that bore sporangia on the adaxial side. The leaf is thought to have folded to enclose the ovules. The folded leaf part formed a valve. In Arabidopsis, since two such carpels fuse, they form two valves. The adaxial tissues lie near the margins of the fused carpels, and these are meristematic and called the carpel margin meristems or CMM. The CMM is responsible for generating the various tissues like placenta, ovules, style, stigma, etc. Four longitudinal major veins run through the pistil, two lateral and two medial. The lateral veins are placed at the center of the valves and terminate at their apical end. They represent the middle vein of a leaf. The medial veins run through the tissue called repulum, which represents the region where the two valves fuse. We shall now see more about the development and details of the structural organization of the pistil. During early development of the Arabidopsis pistil, a single dome-shaped primordium is seen, which is formed by fusion of the two carpels. This lies inside the whorl of stamens. Within this primordium, lateral and medial domains are specified to form the ovary. The lateral domains form the ovary wall or valve. The valve margins eventually develop into the dehiscence zones in fruits. The valves, which represent the lamina of a leaf, exhibit abaxial-adaxial asymmetry. The valve wall consists of an abaxial epidermis on the outer side with small rectangular cells interspersed with immature stomata. Underneath the epidermal layer are chlorenchymatous cell layers representing the mesophyll, followed by another layer of longitudinally elongated cells similar to the leaf palisade cells, and then an inner epidermis that represents the adaxial side of the pistil. The medial domains retain meristematic activity and form carpal margin meristems. The carpal margin meristems of each carpel grow, differentiate, and fuse to form the septum. This is a false septum since it is formed subsequently from the fused carpal primordia. The septum runs along the entire length of the ovary and divides it into a bilocular chamber. The carpal margin meristems also form the placenta within each locule, with ovules arising from the placental tissues. At the center of the septum, there is a transmitting tract through which the pollen tubes grow. The ends of the septum, together with the valve margins from lateral domains, form the rheplum. The rheplum is located at the medial plane, while the valve and valve margin form the lateral domain. The valve margins are made up of a few rows of cells which play a major role in fruit dehiscence by facilitating the detachment of the valves from the rheplum. The cells of the carpal margin meristem remain in a pluripotent state until the pistil elongates and subsequently differentiate into the rheplum, septum, placenta, style, and the stigma. Let us see how pistil development is regulated at the molecular level. As seen in the earlier module, the shoot apical meristem specific Wusch shell activity or Wusch activity and the floral meristem specific leafy activity regulates the expression of agamus or Ag. Now Ag in turn represses Wusch, leading to the termination of the floral meristem. Ag along with sepalata genes play a major role in specifying the pistil. Shatterproof 1 and Shatterproof 2 are two Madsbox transcription factors closely related to Ag, which play a role in early pistil development. Mutants defective in SHP show the absence of the valve margin, which forms a dehiscence zone in the fruit. SHP is expressed over the entire carpel at early stages, but at later stages, its expression is restricted to the ovules and the valve margins. SHP expression is negatively regulated by another Madsbox transcription factor called fruitful or FUL in the valves and by a homeodomain transcription factor rheplum-less or RPL in the rheplum. The full RPL double mutants or the Shatterproof 1 and 2 double mutants give rise to an indehiscent fruit. Now Ag and Shatterproof activate two other transcription factors known as spatula or SPT and crab's claw or CRC. Spatula encodes a beta helix loop helix transcription factor and is expressed in many vegetative and reproductive tissues. In the pistil, spatula plays a role in promoting the development of all tissues differentiating from carpal margin meristem. Hence, SPT mutants show defects in the development of most carpal-specific tissues like the placenta, septum, and the ovules. The crab's claw belongs to the Yabi family of transcription factors and is exclusively expressed in nectaries and carpels. In the developing pistil, CRC expression is restricted to the valves where it specifies the differentiation of tissues that form the ovary wall. The pistil of CRC and SPT double mutants develop as two unfused organs with fewer ovules and less stigmatic and stylar tissues. Hence, agamus, fruitful, rheplum-less, shatterproof, spatula, and CRC or crab's claw together specify the formation of the ovary region of the pistil. At the upper end of the ovary, the style and stigma are derived from the carpel marginal meristem. The style shows a radial organization unlike the bilateral organization of the ovary. It has a distinct epidermis with wax deposits and open stomata, and an internal central core of transmitting tissue which continues into the septum of the ovary. Cells of the transmitting tract are organized into vertical files and produce a complex mixture of polysaccharides, glycoproteins, and glycolipids which form an extracellular matrix. The style also shows extensively bifurcated veins that form what are called as stylar fans. The stigma, which develops at the distal region of the style, is formed by a single layer of elongated epidermal cells called papillae. The stigma is the receptive part of the pistil which functions by binding to pollen grains, promoting their hydration and germination, and providing an entry point for the pollen tubes in their path towards the ovules. The apical basal patterning of the pistil is dependent on an auxin gradient spanning the pistil primordium. Auxin synthesized at the apical end of the pistil is transported basally, forming a decreasing gradient. High levels of auxin induce style and stigma differentiation. Intermediate levels form the ovary, and low levels form the basal gynophore. This was seen by application of an inhibitor of polar auxin transport to the growing apex of the pistil. Inhibition of polar auxin transport reduced the relative amounts of style and ovary growth and increased gynophore growth. In the medial domain, cytokinin signaling induces auxin biosynthesis, and auxin transporters carry auxin to the lateral domains. Likewise, auxin signaling in the lateral domain restricts the cytokinin to the medial domain. Let us see more details about the hormonal gradients which we discussed here. We have seen that the spatula mutants show defects in the carpellary tissues developing from the carpel margin meristem. These mutants also show dramatic alterations in the stigma and style, since both these tissues are also derived from the carpel margin meristem. The carpel specificity of spatula depends on the functionally redundant hectate genes HEC1, 2, and 3, which also encode beta helix loop helix transcription factors like spatula. HEC3 proteins interact with spatula, and both transcription factors play a role in regulating the auxin and cytokinin signals during pistil development, which mediate the style radialization process. The HEC spatula proteins induce the expression of cytokinin signaling components type A ARRs, which antagonize cytokinin function in the lateral domain. They also regulate the PIN protein expression and mediate auxin signaling in the lateral domain. Several genes have been identified that are involved in the style differentiation, but no mutant has been reported that lacks only a style. Hence, the stigma and style seem to be regulated by the same processes. A group of transcription factors called NGATA or NGA 1 to NGA 4 encode members of the B3 transcription factor family and are necessary for both style and stigma development. In the NGA quadruple mutants, there is no development of the style and stigma, and these tissues are replaced by outgrowths showing valve identity. The NGA transcription factors, along with a zinc finger transcription factor called stylish play a role in specifying the styler tissues. Constitutively expressed NGA3 and STY1 plants show a phenotype where valve cells are replaced by styler cells, that is, they show radialization. Both the genes induce expression of the UCA or the YUC genes that are involved in auxin biosynthesis in the apical tissues of the pistil. The auxin is then transported downwards by the pin efflux transporters to generate the auxin gradient as seen earlier. Localized application of exogenous auxin to the apical end of developing pistil restores style and stigma development in the NGA or STY mutants, confirming the role of these genes in bringing about their effects through promoting auxin synthesis. The four NGA genes act redundantly to direct style development in a dosage-dependent manner. Hence, patterning of the style and stigma is brought about by interactions of genes regulating hormone biosynthesis and signaling mediated by transcription factors. Now we shall see how the ovules, which are the sites of sexual reproduction, are formed within the pistil. In Arabidopsis, the ovules arise as small finger-like primordia by divisions in the subepidermal cells of the carpal margin meristem, which form the placenta. Later, carpal margin meristems fuse to form the false septum, such that each valve contains two rows of ovules attached to the inner walls of the ovary near the junction of the two carpels. The placental region represents the site of emergence of the ovules on the carpal margin meristem. The ovule primordium is organized into three different regions along the proximal distal axis: the funiculus, the chalaza, and the nucellus. The funiculus is a vascularized stalk connecting the ovule to the placental region. The nucellus represents the megasporangium, and a cell at the distal end of the subepidermal layer of the nucellus forms the archisporial cell. The archisporial cell enlarges and differentiates to form the megasporocyte or the megaspore mother cell. At this stage, two integuments emerge from the chalaza, which is the integument forming meristematic region at the base of the nucellus. Of the two integuments, the inner integument develops first and grows as a radially symmetrical structure to envelop the terminal nucellus. The outer integument grows asymmetrically, initiating and growing more extensively on the abaxial side of the ovule to envelop both the inner integument and nucellus, and results in an anatropous or recurved shape of the ovule. Let us now try to understand how ovule development is regulated. The class C gene ergamus is involved in both carpel as well as ovule identity, along with the D class genes like the Madsbox transcription factor called seed stick and the E class sepalata genes as seen in an earlier module. Seed stick, along with its homologs Shatterproof 1 and Shatterproof 2, which we have seen earlier, interact with the homeobox protein BEL1 or BEL1 to regulate ovule integument identity and development. BEL1 is a negative regulator of these genes and directs the meristematic cells in the chalazal region to differentiate into integument primordia. The Madsbox transcription factor, sporocytlase or SPL, which was seen to play a role in the specification of somatic and germline cells during anther development, also plays a role in the differentiation of nucellus and the establishment of adaxial-abaxial polarity in the ovule. SPL is expressed throughout the ovule primordium and later is restricted to the nucellus. In the nucellus, it induces the expression of bush shell, which we have studied earlier. However, clavata is not expressed in the ovule, so the bush shell clavata loop, which maintains the stem cells in the shoot apical meristem, is not operational in ovules. In ovules, WUS along with another transcription factor called integumenta or ANT, which encodes an AP2 family transcription factor, is also induced by SPL. So WUS along with ANT contribute to the growth of ovule integuments. ANT is expressed in the chalaza early during ovule development, marking the site of integument formation, while WUS regulates integument formation in a non-cell autonomous manner. That is, though it is expressed in the nucellus, it regulates the expression of genes in cells of the chalazal region. Both WUS and ANT mutants show ovules without any integuments. While WUS and ANT maintain the meristematic properties of the chalazal cells, the BEL1 activity specifies integument identity. BEL1 mutants therefore show the development of a single collar-like tissue instead of the two integuments. Further, the regulation of the differentiation of the two integuments involves expression of two proteins, fabulosa or PHP and the inner no outer or INO. The HD3 zip transcription factor fabulosa is expressed in the early ovule primordia and marks the adaxial side of the ovules, similar to its role in leaf differentiation. Its expression is regulated by WUS and ANT. As the ovule develops, fabulosa expression is restricted to the chalazal region by SPL, where it specifies the inner integument. SPL also regulates the expression of the transcriptional regulator inner no outer or INO, which specifies the initiation of the outer integument in the chalazal region. The outer integument on the abaxial side of the ovule is initiated first, then the one on the adaxial side develops. Hence, the outer integument gives the adaxial-abaxial polarity to the ovule. Once both the inner and outer integuments have emerged, INO expression is restricted to the abaxial epidermal cells of the outer integument. Subsequently, the integuments grow in a concerted manner and cover the entire nucellus, leaving a small opening called the micropyle at the top. The integuments grow to form the ovule wall, leaving an opening called the micropyle at the apical end of the nucellus. The ovules in most species are longitudinally curved such that at maturity the micropyle is located close to the point of attachment of the funiculus to the placenta. The diploid megaspore mother cell undergoes meiosis to form 4 haploid megaspores. Of these, 3 megaspores towards the micropylar end undergo programmed cell death, while the lowermost one is functional and shows 3 mitotic divisions to form the female gametophyte or embryo sac. Hence, Arabidopsis shows a monosporic embryo sac development. After the first mitotic division, the 2 nuclei separate from each other by a vacuole before they undergo the second mitotic division to form 4 nuclei. A third mitotic division further forms 8 nuclei, 3 of which migrate to the micropylar end to form the egg apparatus consisting of an egg nucleus and 2 synergid nuclei. At the opposite pole, Three nuclei migrate to form the antipodal nuclei. The two polar nuclei, one from each pole, lie in the micropylar cytoplasm of the embryo sac and fuse to form a diploid central nucleus. Cell walls are now formed once the 8 nuclei are positioned. This divides the embryo sac into 7 cells with 4 cell types: 3 antipodal cells at the callosal end, a diploid central cell, 2 synergids, and an egg cell at the micropylar end. Hence, the female gametophyte is an 8 nuclear or 7 cellular structure embedded in the nucellus of the ovule, and it bears the single female gamete, the egg. Antipodal cells degenerate shortly before fertilization in Arabidopsis. The synergid cells often lack a cell wall at the callosal end and are sealed by specialized cell wall-like structure called the filiform apparatus at the micropylar end. They also show an opposite polarity to the egg cell. The egg and the 2 synergid cells, along with the central cell, form a female germ unit, that is, a functional unit which is able to attract a pollen tube, interact with the tube to trigger sperm release, and complete double fertilization. Ovule mutants lacking both sets of integuments like BEL1, ANT, and SPL mutants fail to develop normal female gametophytes or embryo sacs besides the integuments. These genes affect both the sporophytic as well as the gametophytic development of ovules. Now, the entry into meiosis requires a reprogramming of somatic cell fate, and this involves repressing stem cell activity. This mechanism involves the cyclin-dependent kinase inhibitors of the KIP-related protein or KRP class. We have seen the role of KRP inhibitors and their removal through the proteasomal pathway, which allows progression of mitosis in the generative cell to form the sperm cells. CDKA is known to phosphorylate the Arabidopsis retinoblastoma related 1 or RBR1 protein that activates the expression of many genes required for DNA replication and hence progression to the M phase of mitosis. KRP-mediated inhibition of cyclin-dependent kinase A prevents entry into the M phase. This inhibition is also important in the formation of a single megaspore mother cell in an ovule. The megaspore mother cell in RBR1 and KRP triple mutants undergo several mitotic divisions instead of the meiotic division. RBR1 is also known to directly repress Moose expression, and hence the stem cell fate of the megaspore mother cell is arrested before it enters meiosis. The functional megaspore divides by 3 mitotic divisions to form the 8 nucleated embryo sac as seen in the earlier slide. RBR1, which we have seen to be important for mitotic progression, also plays a role in restricting the nuclear divisions that lead to the formation of the embryo sac. RBR mutants show supernumerary nuclei in the embryo sacs. Within the embryo sac, a distinct polarity is seen forming the micropylar callosal pole axis. The gametophytic cells are distributed along this axis. Synergids are located towards the micropylar pole and antipodals towards the callosal pole. The egg cell is in the domain adjacent to the synergids, and the central cell lies in the domain adjacent to the antipodal cells towards the center. Each type of cell communicates with its neighbor. The micropylar callosal axis is maintained by auxin, which shows a maxima in the nucellar cells adjacent to the micropyle. The cytokinin insensitive 1 or CKI1, which is a cytokinin signaling histidine kinase, is expressed in the callosal nuclei that form the antipodal cells and also in the central cell where one of the callosal nuclei migrate. CKI1 mutants show a loss of antipodal and central cell identity and acquire an extended synergid and egg cell identity. Another mutant that shows defects in the micropylar callosal axis is the altered meristem 1 or AMP1 mutant. AMP1 encodes an endoplasmic reticulum membrane located a carboxypeptidase and is an inhibitor of cytokinin synthesis. AMP1 is expressed in the integuments during early gametophyte development and later it localizes to the synergids where it promotes the fate of these cells. Hence, the micropylar callosal axis is formed by auxin and cytokinin signaling mediated through the AMP1 and the CKA1. Superimposed on this micropylar callosal axis, several cell autonomous factors specify the formation of the different cells of the embryo sac. A MIP transcription factor, MIP98, specifies the synergids and its mutants lack the filiform apparatus and instead acquire the egg cell fate. However, its role is not yet fully understood. A RWP-PK domain containing transcription factor called RKD is a plant-specific transcription factor which has a characteristic RWP-PK DNA binding domain and specifies egg cell differentiation by regulating a large number of downstream genes. RKD mutants have an egg cell showing synergy-like features and its constitutive expression induces the formation of additional egg cell-like structures within the embryo sac as well as in the surrounding sporophytic tissue. Finally, the AG-like or the AGL80 and AGL61 genes encode Madsbox transcription factors which specify the central cell. Their function is also not yet completely understood. Antipodal cells degenerate shortly before fertilization. So the female germ unit is formed by the synergids, the egg cell and the central cell. We now come to the end of this module in which we dealt with 3 phases in carpel development. In the first phase, we dealt with the common primordium of 2 fused carpels that forms the pistil and how it develops into the stigma, style and ovary. In the second phase, we saw how the ovule develops within the pistil and the formation of the megaspore mother cell within the nucellus of the ovule. Finally, in the third phase, we studied how the megaspore mother cell undergoes meiosis to form the haploid megaspore and the subsequent mitotic divisions that lead to the formation of the female gametophyte or embryo sac in which the female gamete, the egg, is located. In the next module, we shall study the processes of pollination and fertilization which bring about the union of the male and female gametes. Thank you.